Hi, this is Philip from the Amphasys support team. Welcome to today's video session on how to run a sample using the Ampha Z32 impedance flow cytometer. In a previous video, we learned how to prepare a sample properly, so now we want to measure it. Please note that I am using an official Amphasys template. Templates. These templates can be downloaded from our website amphasys.com from the download section. Templates are available for normal mode or counting mode measurements. They come pre-configured for a broad variety of species. For species not listed in the package, please contact support@amphasys.com. Once downloaded, we need to unzip the template package and select the folder, or as we call it from now on, the workspace, named after your species of interest. Each of these workspaces comes pre-optimized with all the necessary settings already selected for your convenience. After opening the workspace in Amphasoft, you just need to enter the ID of the chip that you are using as well as your individual sample names. Please note that the necessary connection between computer and instrument, as well as the rinsing of the fluidic system, have already been completed in my setup here. For a better overview, we split up the video content into three phases, which are pre-measurement, during measurement, and quality control. First phase, pre-measurement. The necessary preparation steps are renaming samples and nodes. Renaming samples and nodes can be done either from the measurement window or from the sample list. The entries are consistent in both directions. There is also the possibility to import sample names and nodes from an external file using the import function. With the import function, you can add sample names and nodes from a CSV file into your workspace. To demonstrate that, I prepared such a CSV file for you. Sample names have to be in column A, nodes in column B. All other configurations, such as buffer ID, stop conditions, or alike, are not possible to configure with this function. To be able to import sample names and nodes, you need to delete all but one measurement from the sample list of your workspace. The next step in preparing a measurement is the sample preparation. To learn how to properly do that, please check out our separate tutorial video on this very topic. Second phase, during measurement. Now it is time to start the measurement of the sample. Start measurement. To start a measurement, click the button Start Measurement at the lower end of the measurement window. Please note that by standard, the first phase that will follow is a loading phase as indicated by the status window on the lower left side. This phase is in place to minimize the waiting time that is necessary for the sample to reach the chip where the actual measurement will happen. Now we see the data from our measured sample starts to appear in the measurement window. To stop a measurement again, you can either use the pre-configured stop conditions, which will automatically terminate a measurement either after 2 minutes or 10,000 measured cells. Please note that these stop conditions can be customized according to the requirements of your experiment. Alternatively to the stop conditions, or in case you need to stop a measurement immediately, you can also stop the measurement manually by pressing the same button with which you started the measurement. Please note that by default, this will cause a flush where water from the water bottle is used to clean the entire fluidic system to prevent sample carryover. Should you stop the measurement due to a chip clogging, please note that you should press the same button once again to also end the flush function. Third phase, quality control. Let's have a look at the data we just measured. There are mainly two criteria to assess the quality of a measurement. Rejection rate. The rejection rate describes a percentage of all accepted cells. If signals do not fulfill the quality criteria of our trigger algorithm, they are filtered out, not appearing in the scatter plot and not being counted. It is displayed here at the lower right side of the screen. If the rejection rate in general stays below 5%, it's a sign of a good data quality. Any percentage above 10% does indicate a suboptimal setup, and we do not recommend to use such samples. Separation between dead and living populations. In an ideal case, the separation of living and dead cells can be seen on the first glance as two completely separated populations. The viable cells appear further right on the screen. The non-viable cells appear further to the left. Let me explain what the axes of the scatter plots show and what their meaning is. Y-axis. The y-axis shows the amplitude. The unit is in volt. Dependent on the applied frequency, it indicates the cell surface, which can be seen as an equivalent to the relative size of the cell, or the membrane capacitance and conductivity of the cytoplasm, x-axis. The x-axis shows the so-called phase shift. The unit is in degrees, the same as in an angle or a circle. A viable cell, because it still has metabolic activity, causes a phase shift that is much more pronounced than the one of a non-viable cell. Alright folks, that's all for today. Thanks for watching and see you next time around. Your Amphasys support team.